Welcome to CET Health 25. I am Srilakshmi Nair and today I am going to explain the topic microsporogenesis. So, we will get into microsporogenesis. First, we will recall what was taught in the last class. What is microspore? So, microspores are small structures. It is found in the male reproductive part of the plant, that is the anther. We can find mature microspores in the anther. So we had come across the term sporogenous tissue, which is present within the thecas of the anther. So this is depicted. It shows the sporogenous tissue which is present inside the anther. So we can see that these are the cells which are the sporogenous tissue, and these are not mature. It is yet to form into a mature pollen grain. So that is the formation of a microspore into a microspore tetrad is known as microsporogenesis. The sporogenous tissue or the microsporangium contains multiple cells. These cells undergo meiotic division. Meiotic division, as I had mentioned earlier, it is the division, it is the splitting of the cell into two parts, where the actual resources within the cell gets divided into two separate cells. That is, there is a formation of cell wall within the cells. So, so here the sporogenous tissue cells or the microsporangium undergo meiotic division to form four cells in one, which is known as a microspore tetrad. So, the microspore tetrad disassociates. It dehydrates itself and it disassociates to form into four pollen grains, or these are a potential pollen mother cell. Each of the cells dissociate from the microspore tetrad is a potential pollen grain. So I have depicted four of them, but this shows the maturation of one of the cells. So this PMC, potential pollen grain, this pollen grain forms a air vacuole within itself and it undergoes mitotic division. This pollen grain, which is dissociating from the microspore tetrad, it forms a food vacuole or a air vacuole within itself, and it divides itself and splits into two cells within itself. Mind you, this cell does not split into two different cells, but it stays as a two-celled stage. This two-cell stage is a mature pollen grain. So what we can see here is that there are two cells here, and two of them form a mature pollen grain, and this is the final mature pollen grain or a male gametophyte. In the final pollen grain, we can see that there are two cells within one cell, that is the vegetative cell. The bigger cell is a vegetative cell and it performs the function of storing food. It is a food reserve cell. Is the generative cell. This generative cell is a small cell compared to the vegetative cell, and it is spindle shaped. That is, the shape of the cell is small and it forms a spindle-like structure. The generative cell in fact contains a more dense cytoplasm. This is the mature pollen cell and it is a two-celled structure. Thus, I would like to conclude my session. I hope you understood the topic of microsporogenesis, that is the maturation of a sporogenous tissue or microsporangium into a mature pollen grain or a male gametophyte. Thank you for watching. I hope you liked the video, and if you have any comments or any suggestions, please put them in the comment section. Thank you.